Hi, my name is Rafael Canevarolo. I'm a research scientist at Dr. Arosto Silva at Moffitt Cancer Center, and today I'll be presenting my work called Multiomic Landscape of Evolution of Warburg Effect Identifies Drivers of Breast Cancer. So in this work, we study MCF10A, which is an epithelial non-tumorigenic breast cell line, in which we put the cells into repeated cycles of nutrient starvation. After 18 cycles of starvation, we split and expanded these clones. So we were able to identify high and low Warburg effect clones. And we performed several experiments, whole exome sequencing and single cell multiome on these Warburg effect clones. We are able to identify several copy number alterations in these Warburg effect clones, mainly associated with reactive oxygen species, ROS, using cosmic signature. We are also able to identify, for example, focal amplification of the MYC locus on chromosome 8q in all of the clones. Also, we could track all the chromosome loss and gains and specific mutations in each of these clones, thus reconstructing the phylogenetic history of each of them. Later, we could perform single-cell multiome on these cells. So we had paired RNA-seq and ATAC-seq data for all of them. And surprisingly, we noticed that the clones would not cluster according to their Warburg effect status, but actually according to their epithelial and mesenchymal status. We could also investigate different pathways associated with each of these phenotypes. And we noticed that for each of these clones, we had epithelial and a mesenchymal compartment. So, for example, we could identify specific pathways associated with the mesenchymal phenotype and the epithelial phenotype, just like mTOR signaling in all of the clones, but MYC targets mostly in the epithelial compartments. We also found differential transcription factors that could be regulating this abnormal gene expression. Lately, we could, by correlating gene accessibility, target accessibility with gene expression, we could pinpoint putative pioneer transcription factors for breast cancer that deserves further investigation. So, as a conclusion, we hypothesized that actually is mixture of mesenchymal and epithelial phenotypes in each of these clones that ultimately give us what we call high or low Warburg effect status. Moving forward, what we can do is to investigate these pioneer transcription factors, their specific roles in determining DMT transition, as well the Warburg effect. And we show here that both environmental and molecular aspects of the clones are very important to determine the Warburg effect as well as DMT phenotype. Thank you very much.